Finally, tonight, we have Ronan Carroll from Biological Sciences, and he's going to be talking about vaccine denial, facts and fiction. Ronan, it's all yours.
Thanks, Sarah. It's great to be back. This is the second Science Cafe that I've given, and the last time I talked about antimicrobial resistance and the rise of antimicrobial resistant bacteria, but that wasn't controversial enough, so I wanted to come back and do it properly. So tonight we're going to talk about vaccines. We're going to talk about what vaccines are, we're going to talk about how they work, and then I'm going to talk a little bit about some of the controversies surrounding vaccines. So before I get started, can I just get a quick show of hands? Hands up anyone who got their flu vaccine last year. Okay, that's a really pretty low percentage, all right? Hands up anyone who got the flu last year. Okay, not too many, but a few, okay. So I did both, right? I got the flu vaccine and I got the flu. And so this is a common thing that we get, is anyone who got the vaccine and then they get the disease, they're like, well, those things don't work. And that can lead to sort of some of the arguments that we're going to talk about tonight. So hopefully I can explain to you a bit more about what vaccines are, how they work, and then we'll sort of discuss some of those controversies as I move along. So let me see if my clicker is going to work. Here we go. So the numbers have pretty much come out now on last year's flu season, and the 2017-18 flu season was one of the worst that we've had. Okay, you've probably seen reports in the media, reports that look like this, kind of outlining how bad the 2017-18 flu season was. I think it's been estimated that it's the worst flu season since about 1976, so the worst one in a few decades, right? The CDC has estimated that there were about 900,000 hospitalizations, which led to about 80,000 deaths in the flu season last year, right? Just take a second to think about that. 80,000 deaths is a huge number of deaths that are either resulting from this infection or from complications arising from that infection. And unfortunately, about 180 of the deaths that are associated with that were amongst children. This is the highest number of children fatalities since the CDC started taking records for influenza a couple of years ago. So this was a really, really severe flu season that we just went through. And there's two hallmarks of last year's flu season that really kind of stand out and make it different than most other years. The first is the age groups that were infected. And so typically what we see every flu season, we see high rates of infection in people over 65 and amongst children and toddlers, right, and babies. These are the demographics, these are the age groups that typically suffer from the highest rates of infection. Well, last year, we saw high rates of infection in all age groups, right? So people in their 30s and 40s were also getting the flu, and that's something that you don't normally see. So that really is a hallmark of an extremely high activity flu season. The other thing that we saw last year was high activity throughout the United States. And so again, we tend to see sort of regional clustering. You tend to see more flu in northern states than you do in southern states. But last year we saw flu very widely distributed throughout the United States. And there were a couple of weeks, I think there were 6 weeks back to back, where there was high flu activity reported in all 48 continental United States at the same time. And that's not very common. That doesn't happen except in the worst of flu seasons. So a really, really bad flu season. This chart is taken from the CDC, where they've published some of the data about the numbers of infections throughout the flu season last season. So let me just walk you through some of the information that's on this chart. What you're looking at is a week-by-week number of confirmed reports of influenza in the United States. And so you can see that for each bar, there are two separate parts. You've got the yellow on the top, you've got the green on the bottom. These indicate the number of positive influenza A and influenza B tests that were reported. Okay, so you can see from this that straight away there's two types of influenza, A and B, and I'm going to come back and talk about that in a little bit. What you can see is influenza A is pretty much the more common one, influenza B less common, but it tends to come on a little bit later in the season. You start to see influenza B infections later in the flu season. So just so you can orient yourself, because I know you probably can't see the numbers down the bottom here, I've kind of marked with the blue arrow here where the December-January switch is. So that's week 52 of the year. And so you can see this at the end of December, beginning of January is where we're really starting to see these numbers of infections peaking. But the infections start much earlier than that, okay? You can count backwards. And the first week that's shown on this graph is week 40. Does anybody know what week of the year we are in right now? Anyone? 39, okay? So we're in week 39. We're 1 week before this. And so while the numbers of infections are still relatively low for these first few weeks, we are really at that pivotal point. Right now is the time to get your flu vaccination so that you don't become one of the numbers that's on this chart. Another interesting aspect of last year's flu season is you can see that by the time it declines and really finishes, you're talking about late May 2018. So that's a much more extended, that's typically longer than what we would normally see for the flu season. So again, just kind of highlighting how many cases that were seen by the CDC last year. So I'm going to break this talk down tonight into 2 talks because you're not getting a science cafe next week. I'm going to do 2 science cafes tonight. Right, so the first talk I'm going to give you is I'm going to talk about vaccines, what they are and why do they work. Okay, I'm going to outline exactly the immunological reason why we use vaccination and how it works. And then the second thing I'm going to talk about towards the end of my talk is vaccine controversy. I'm sure everybody in the room has heard about some of the controversies surrounding vaccines and their use. It's probably the reason why half of you are here tonight to hear what I have to say about it. Before I get involved in any of this, I would say I'm more than happy to be interrupted as I talk. If you disagree with something that I say, I would really love to hear from you. It's also a way to get in early and get that t-shirt before people start asking questions towards the end. So if you ask a question during the talk, you still get the t-shirt. Right, Sarah? Yes, okay. So first off, what are vaccines and how do they work? And so to talk about what vaccines are and how they work, we really have to consider the human immune system. We have an absolutely fantastic immune system, each one of us, right? It's a series of molecules and cells and organs inside your body that are dedicated to fighting disease and fighting infection. And your immune system can contribute in the fights against cancer and other types of diseases, but what I'm really interested in is how your immune system fights against infectious diseases that are caused by bacteria and viruses. Because we now know every day of your life, you are exposed to hundreds of millions of different microbes, right? And so your immune system is what allows you to fight off infection and not contract disease when you're exposed to these microbes. So if we look at the human immune system, we can break it down really into 2 different parts. There's 2 parts to your immune system. The first part is what we call the innate immune system, and the second part is what we call the adaptive immune system. And I want to take a few minutes just to tell you some of the key characteristics about these 2 different parts of your immune system, for those of you that are not familiar with this. So starting off with the innate immune system, your innate immune system is a non-specific defense mechanism. It's a non-specific way to fight against infection. And we think of our innate immune system really as being the first line of defense against any kind of infection. A really good example of something that's part of your innate immune system is your skin, right? Skin is probably the most important part of your innate immune system because it keeps things out, right? Simple as that. Your skin keeps microbes out of your body. If you can keep the microbes out, they can't get in and they can't cause disease. So we think of really basic barriers like the skin and the mucous membranes as being part of your innate immune system. But we also have more complicated things inside your body, the inflammation process. When you get an infection, you get inflamed. That's all part of your innate immune system responding to that infection and trying to clear it. We have phagocytic cells. These are cells inside your body that can eat bacteria and viruses and destroy them. And then we have a variety of antimicrobial substances that are produced that are there to try and kill these invading bacteria and viruses. Our adaptive immune system is our second line of defense. And there's a variety of different molecules, cells, and organs inside your body that are part of the adaptive immune system that also contribute to the ability to fight infection. And so when I'm talking about these 2 parts of your immune system, there are 3 very important characteristics that I want to mention that really differentiate between these 2 parts. The first is the speed with which these 2 parts of your immune system can respond to an infection. Our innate immune system is very fast, and it can react almost instantaneously to any infection. And the reason for this is that all of the components of your innate immune system are already in place within your body. They already are there and they exist. You have your skin on all day, every day, right? You don't take it off. You don't have to make skin when there's a bacteria present, okay? So that's the way to think about the innate system. It's always there. It's constantly ready to fight off any intruder, okay? In contrast to that, our adaptive immune system is quite slow, and it takes time to respond to any infection. And we'll come back and show why that's important later on. The second aspect of our immune systems I want to talk about is specificity. And the names up here, I think, already give this away. Our innate immune system is not very specific. And what I mean by that is it's active against a wide variety of bacteria and viruses. A wide variety of microbes can be targeted and killed by the innate immune system. In contrast to that, the adaptive immune system is very specific. It may be specific for a bacteria. It may be specific for a very particular type of bacteria. And so it's much, much, much more focused in the range of microbes that it can be active against. And the final part, and this is the one I really want to talk a little bit about, is memory. And whether or not these two branches of our immune systems have memory. And what I mean by that is whether or not these parts of your immune system can remember an encounter with a microbe and how that alters its response in the future. Your innate immune system does not have any memory. It doesn't remember encountering any microbes. And so what that means is that every time it encounters the same microbe, it responds the same every single time. The strength of the response is not altered. The first time, the second time, third, fourth, fifth time you encounter the same invading microbe. In contrast to that, your adaptive immune system does retain memory. And what that means is the second or third or subsequent times that you encounter a microbe, the response of your adaptive immune system will be quicker and it will be stronger. And that enables you to clear an infection in many cases before that infection even gets a chance to establish. Okay? And so it's this aspect of your adaptive immune system that is the key aspect on which vaccination is built. Okay? Yes, first question. How is the memory retained?
You said like it's like the responses has to be made. So if it's not like saved in there, like how does it build on it?
So the question is, how does this memory come about? Where does it come from? And I promise you in about 4 slides, I have a slide that's going to go some way to answer your question. So is that okay? Can I do it when I get there? All right, perfect. So, another question.
Are different people born with like different innate immunities? Are different people born with different innate immunities? And does it vary throughout the population?
Are different people born with different innate immunities? I'm sure there's some variation person to person, but in general, I think the innate immune system is thought to be somewhat consistent across people, whereas the adaptive can be a lot more variable, I would think. Yes?
Is there like a limit to how long your adaptive immune system can like remember?
Is there a limit to how long your adaptive immune system can remember an encounter? That's a very variable question. In some cases, yes, the memory might be over weeks or months, but then goes away. In some cases, no, it can be extremely long-lived and be around for the rest of your life. Yeah, good question. Yes?
I have allergies, and I get worse and worse reaction to allergies over time. How does that jive with what you're saying about the system?
Yeah, so allergies kind of sort of fall outside of that realm. Allergies, I think, is typically an over-activation of the innate immune system. It's my understanding of it. And so that's really, the allergy, what you feel is really the immune system's response to whatever the allergen is that's causing that. Does it get worse over time? If it does, then perhaps there is some adaptive immune component to that as well. It would very much depend on what the allergen is and the type of allergies that you're talking about. But it could do, yes. Yes?
Could the same microbe that goes into the innate immune system react differently inside? Or are they two different microbes that you're talking about?
So when it comes to these, so it's convenient for me to discuss the innate system, the adaptive system as being separate entities. The reality is they're not. They're really part of one large system, and there's a lot of bridging between the two and crosstalk between the two. But kind of for simplicity purposes, it's easy to discuss them like this. But any bacteria, any microbe in the body will be recognized by both branches, and both branches will respond to it. In certain cases, the innate immune system may be sufficient to clear it. In other cases, you may need help from the adaptive system to fully clear that infection. Yeah. Yeah.
What happens if you have a skin condition that allows breakage in the skin or the skin falls off easily? Would the innate still be first or would it switch to the adaptive?
So the skin is just one part of your innate system. There's lots of other parts. There are different cell types. There are different molecules. Those may still be produced even if you have some skin breakage or some skin deficiencies. So you might just be defective in one aspect of your innate system, but you have all the other components. Yeah. Yeah.
What exactly causes seasonality of the influenza and causes it to happen in season?
What causes the seasonality? So the seasonality also seems to be linked to temperature, right? That seems to be a huge aspect to that. So cold temperatures seems to facilitate the spread of the virus. I'm not sure if there's much known about the molecular reasons for that. If the virus seems to survive better in cold temperatures, that could be a reason. But definitely there's a link between temperature. So we already know that the flu season, it has started in Australia right now because they're in their winter. And so we often get indications from that. But what causes it to follow the seasons in terms of temperature, I'm not really fully sure of myself. Yeah. Yeah. Yes?
So my question is related to skin. So would it be any difference between having thicker skin, a thinner skin, or color of the skin?
I mean, when I'm talking about skin here, I'm talking about it in very basic terms in that it keeps stuff out of your body. But, you know, yeah, there's so many different skin types. I'm sure that there are differences in how well it works in terms of protecting. But, you know, just on a very simple level, on the one hand, no matter how good your skin is, it still keeps a lot of microbes out of your body, right? Yeah.
Isn't there four types? Is it passive one too?
4 types of... Oh, you're talking about passive immunity? Yeah, so passive immunity is the idea that...
Repeat the questions.
Oh, apologies.
Question over here too, when you finish that.
Okay. Isn't there another type of immunity called passive immunity? So passive immunity, I think, refers to the idea that a mother can pass on antibodies to her baby. And so because antibodies would be considered part of the adaptive immune system, I would consider it a sort of a subset of that, if you like. Okay? Yes?
Right, this sounds like a stupid question, but like,
No.
Can there be like mutations in the immune system, and if so, like how does the body like kind of try and counteract that to like keep us from like getting sick?
So the question is, can there be mutations in the immune system, and what does the body do to counteract that? Yes, there can. There's a number of genetic conditions that are known where people can be deficient in different aspects of either the innate system or the adaptive system. And by and large, what you find is that those people are more susceptible to infection. Depending on what the mutation or what the defect is, it can be infections by a certain virus or a certain bacteria. Again, just very much depends on the specifics. Yeah. Yes?
How would the body react to something that uses our immune system against us, something that hijacks?
How would the body react to something that uses our immune system against us? How would the body react, or how would the microbe react? The body? Okay, so on the one hand, you have autoimmune diseases, which is the body's immune system targeting itself. And so those can be quite debilitating, and it's kind of hard to treat that without basically repressing the immune system to prevent that. And so how does the body react to that? I mean, I'm not sure I understand what you mean.
I just meant, how would you deal with something like that?
It's difficult. It's very difficult. If the immune system is working against you, there's a lot of very severe diseases that can result in. And really immune repression is the only way to kind of deal with that, try and weaken the immune system, which of course then can have its own problems. Yeah. My goodness, questions everywhere. Yeah.
You mentioned that usually the flu is most common in elderly. Why isn't it children? Since it seems we hear more about children getting flus like during school season. So is it because their immune system breaks down as you get older?
Yeah, so the question is, why is the flu more common in the elderly and in children? And yeah, it simply comes down to how robust the immune system is. So as you age, your immune system becomes less active, which means you're more susceptible to infections. And again, babies and young children whose immune systems haven't fully developed are also susceptible for just that exact reason. Yeah. Yes?
If you're already like immune to something, but then whatever that is that you're immune to like mutates, kind of like the diseases now, if they kind of change, does your memory still remember that you're like immune to that, or do you have like another shot?
Yeah, so the question is, if you are immune to something and you have memory of it, if that mutates and changes, do you still have memory and are you still immune to that? And the answer is no. And that's the reason why there's a new flu shot every year, okay? And so I have a couple of slides on this later on that I don't think I'm going to get to.
You can take a break in questions and move a little.
All right. Maybe we'll take a break in questions and I'll try and get there. But yeah, so the flu virus is very good at mutating and changing. And that's why the vaccine that you get this year may not be efficient, maybe not efficient next year, okay? All right, so let me kind of blow through a few more slides here, and then I'll stop for a few more questions. So I wanted to talk a bit more about this immunological memory of our adaptive system and how that works in terms of vaccines, right? And so there's one molecule I really wanted to discuss, and those are antibodies. And I'm sure most of us have heard of what antibodies are. Antibodies are these proteins, and I don't know why I'm skipping ahead here. These are proteins that are produced by our adaptive immune system. Okay, and this is a simple diagram of what an antibody looks like. And up at the top here on our antibody, we have these two regions that we refer to as antigen binding sites. Okay, and what happens is these antibodies can bind with a really, really high affinity. It's a very, very tight binding to a molecule called an antigen. And what an antigen is any molecule that's recognized as being foreign by your immune system. So the immune system is very good at recognizing your own body and then distinguishing that from something that's foreign, something that comes from outside of the system. Okay? And so an antigen could be an entire microbe, it could be a bacteria or virus, or it could be just a small part of a bacteria or virus. And so our bodies produce these antibodies that can bind very, very tightly to these antigens. And that's very important in terms of how we fight infection, and I'll kind of show that over the next few slides. One aspect of this that I find absolutely, truly fascinating is that if you look at this in detail, it's been estimated that our bodies could make somewhere in the region of about 10 trillion different antibodies that could recognize 10 trillion different antigens. That is an enormous number of antigens. And so it's been estimated that really no matter what molecule there is out there, no matter what bacteria or virus or molecule that's on their surface, our bodies technically have the capability to produce an antibody that could recognize that antigen. And this is very, very important for the principle of vaccination. So, how does antibody production work? If we have an invading bacteria or a virus, it's got antigens on its surface. So there's an antigen that's present on this bacterium. And so there's a type of cell in your body called a B cell. It's part of the adaptive immune system. And it can recognize this antigen on the surface of the bacterium. And when it does so, it starts to rapidly divide. And when that B cell divides, it starts to produce cells called plasma cells. And these plasma cells start to then rapidly produce antibody that recognizes whatever antigen it was that was detected on the surface of that cell. And so as these plasma cells start to produce these antibodies, they flood them into your bloodstream, they flood your body, and eventually those antibodies are going to find and bind to the antigen on the surface of that bacteria. Okay? And now I'll come back to what that does in a minute, but I'm going to go back and answer this gentleman's question next. A very small portion of these B cells, instead of becoming plasma cells and making antibodies, they turn into what we refer to as memory B cells. And these memory B cells can make the same antibody that recognizes this antigen, but they are essentially dormant and they lie primed, ready to go if this bacteria and this antigen ever appears in the body again. So once it gets cleared, these memory B cells are retained, and if it ever experiences this same antigen, this same bacteria again, these B cells can rapidly start producing antibody and clear that out of the body before the infection even gets a chance to set up. Okay? And so what is so great about antibodies? How can they help us fight infection? And so once these antibodies are produced, they basically find their antigen. They find the virus or they find the bacterial cell that they can bind to, and they do so. And when they do so, it really allows our immune systems to clear these bacteria and viruses very, very quickly from your body. So once you start producing antibody against these antigens, the rest of the immune system comes in and cleans these out and gets rid of them. And that's the basic principle behind why antibodies are so effective. Now, I've talked to you about the response and how the adaptive immune response is pretty slow, and that's true. This chart is kind of showing antibody production after exposure to an antigen. So you get exposed to an antigen here, and then it takes 1 to 2 weeks before you really start to produce antibodies. So this is a relatively slow process. But the key thing here, and this is where vaccination is important, a second exposure, I don't know why my slides keep jumping forward, I apologize. A second exposure to the same antigen, which happens here, you see a much faster response. In this case, by about 1 week, you're seeing a much, much greater response than you saw the first time round. And so you see a quicker response and you see a stronger response. And usually this second response is enough to clear that microbe from the body before the infection even sets up. And this is the basic principle on which vaccination is built. So a vaccine is a preparation of microbial antigens that are injected into the body. You then develop antibodies against them so that the next time you are exposed to those same antigens, your immune system can clear up the infection before it even starts. Okay? I'll take a break for any more questions if anybody wants to throw something. Yes?
Where are the B cells produced at? Like,
Where?
Yeah.
Bone marrow. Where are the B cells produced? They're produced in bone marrow. Yeah, hence the B. That's how I remember them. Yes?
When you say that the cells that are produced lay dormant, does that mean like they're throughout all of your body cells, or are they in one specific location?
The cells that are produced, the B memory cells, they lay dormant. Yeah, there is a small number of them that are retained and they're constantly circulating throughout your body, ready to fight this infection if it ever comes back. Yeah. Yes?
Do memory B cells act as normal B cells ever?
No. Memory B cells will only ever respond to that same antigen again if it appears. Yeah.
Yeah, sorry, I'm still.
All right. Hi.
Does those graphs change like every time you have an exposure, or does it mostly remain fairly similar to the second exposure?
In most cases, it will get slightly better each time. Eventually, so the question is, will the graph get better every single time, second, third, fourth, fifth exposure? It generally gets better second, third, fourth, but it will plateau, and eventually it gets to a point where it doesn't get much better. But yeah, there can be an improvement from second to third, and that's why we often get booster vaccinations, right? So one vaccination will generate this response, and a second booster is required to get it up, and then a third one, and then eventually you've reached the point where you're protected. So sometimes it takes a couple of shots. Yes?
At the beginning, you mentioned adaptive immune system is specific, but we only have IgA, D, E, G, M. How do you determine the specificity? Because we only have 5 Ig.
Sorry, can you repeat your question?
Like at the beginning, you mentioned the response of adaptive immune system.
Right.
Specific.
Yes.
How to determine this specific? Because we only have those Ig proteins.
Right, so there are 5 major classes of immunoglobulins, which are different types of antibodies, but the potential antigens that they can bind to are not limited to just 5 different types. The antigen binding domain, which is the part that actually recognizes the antigen, that's not limited to just 5 things. There are, like I say, about 10 trillion different possible combinations. And that's a very, very detailed aspect of immunology. You can take my class in pathogenic bacteriology if you'd like to learn more about that, or the immunology class. But that is definitely a little bit more detail than I want to go into today. Yes? Question?
So kind of go off that, IgM is the first antibody produced.
Right.
So when does it differentiate into the other types?
So when does differentiation... Oh, sorry. So the question was IgM. So there are 5 different types of antibodies, right? They go by different letters: IgG, IgM, IgA, IgE, etc. And IgM is usually the first one that's produced, but then that switches over at some point to IgG. Again, there's a well-defined process for that. It has to do with combinatorial splicing between VDJ segments. Again, it's quite detailed, but when does it happen? It would depend on the specific antigen response, to be honest. But again, my pathogenic bacteriology class is a great place to discuss this more. BIOS 4230, all right? Yes?
Since there are 10 trillion or so combinations for antigens, are those like determined randomly, or how does the first one come about that can bind to a pathogen?
So again, there's a lot of different ways that these different antibodies can be produced, right? And it's a fascinating area of biology. To be honest, if I wasn't a microbiologist, it's probably what I would be studying. But to get these, I could go back to that simple diagram of an antibody where you have that kind of Y shape, but you actually have 2 proteins that come together to form the antigen binding domain. And there is a large number of different segments of genes that can get spliced together to make those. So there's a VDJ segments that can come together, and you can get hundreds and thousands of different combinations. But then there's also a system whereby if an antibody can recognize an antigen, and it's not a particularly strong interaction, right? It's a pretty weak one, it's not very sticky, but that over time and over repeat exposures, the gene that encodes that antibody can mutate and get better. It's truly fascinating how it does this, and there's feedback mechanisms so that the cell knows that certain mutations are actually better, and it selects for those. And so those memory cells can actually get better over time, and the antibodies that they make can get better over time as well. Okay, great questions. Yes?
So
So is your question referring to like the to the antibody production? So is there any evidence that this can get passed on in an epigenetic fashion? Typically, no. A mother can pass on preformed antibodies to her fetus or to her baby. But a lot of this combinatorial splicing and how we develop antibodies from B cells actually happens within your own body. Yeah, because it's a terminal differentiation. And sorry, I know there's a lot of like technical jargon, but once a memory B cell goes down the path of being able to produce a specific antibody, the splicing that happens means it can never go back. And so it's locked into that path and it will only ever be able to detect that particular antigen ever again. Yeah. Okay, let's keep going. So the next thing I was going to talk about is what we put in a vaccine, right? And so there's basically two different types of vaccines. One is where we use either whole cells or whole viruses, and that's what's put into the vaccine. Or the other is what we call a subunit vaccine, where we just take a small part of it. We just break off a piece of that bacteria or that virus, and that's what's actually used as the antigen in the vaccine. And so the first type are whole cells or whole viruses, and as the name suggests, the vaccine consists of the entire cell or the entire virus. But obviously that doesn't make any sense. You're not going to inject somebody with a bacteria or a virus because that would get them sick, right? So how do we get around that? Well, we get around that by either one, using inactivated or killed bacteria or viruses. The other option is we can use attenuated strains, and those are live strains, but ones that don't actually cause disease. Okay, and so many of the vaccines that you'll be familiar with are whole cell or whole virus vaccines that are either inactivated or attenuated. The other type, as I said, are subunit vaccines, where you can purify a molecule off the surface of a bacteria or a virus and inject that. That serves as the antigen, and you can raise antibodies against that. Okay, so at this point in the talk, I was going to switch over and start to talk a little bit about the flu and ask the questions. Well, what do we use for the flu vaccine? And then why do we get a flu shot every year? But thankfully I had a question from over there, so I think I've already addressed that. But nonetheless, I'm going to keep going and talk a little bit more about the flu or influenza, if my slides will behave. All right, so the disease is influenza. We call it the flu. It's caused by a virus, the influenza virus. And this is one of the most successful viruses of all time at killing humans. This thing is really, really good at killing us. The first outbreak of the flu dates back to Hippocrates in 412. Well, that's the first recorded outbreak. Obviously, it had been happening for a long time before then. And it's been estimated that influenza is responsible for about 31 global pandemics. And what a pandemic is an outbreak or an epidemic that spreads throughout the entire world, okay? And so it's been estimated that there are 31 documented pandemics caused by the flu. The first in 1580, but by far the worst pandemic caused by the flu was in 1918, which is the pandemic known as Spanish flu. This has been estimated to kill somewhere in the region of about 50 to 100 million people in 1918. To put that in context, World War I, which had just finished, has been estimated to have killed somewhere between 12 and 15 million people. And so the year after World War I, we have this flu epidemic killing somewhere in the region of 50 to 100 million people. So again, a particularly deadly disease. If we look at the influenza virus itself, we can break it down into, there's 4 different groups: influenza A, influenza B, C, and D. But as I mentioned at the start when I put up this chart, the vast majority of infections in humans are caused by influenza A, followed by influenza B. C doesn't really cause many diseases in humans, and D only infects dogs. So we typically don't get many infections in humans caused by influenza D. As you all know, it's acquired by inhalation. When somebody coughs, you can inhale the virus as they expel it. I want to take a minute to look at the surface of the influenza virus. And this is a cartoon depiction of what that surface looks like. And there's 2 very important proteins that are present on the surface of this virus. And this goes a little bit to understanding why we have to get a new flu shot every year. These 2 proteins are called hemagglutinin and neuraminidase, and they're colored red and blue on this slide here. There are 16 different HA types, and there are 9 different NA types. And so we can classify the flu virus based on which particular HA and NA type it has. So if, for example, this virus has hemagglutinin type 1, neuraminidase type 1, we call it H1N1. If it's got hemagglutinin type 3, neuraminidase type 2, it's H3N2. And I'm sure many of you, I see nods around the room, have heard of these H1N1, H3N3 kind of things. And actually, these 2 viruses are probably the most common ones that circulate every year. But this kind of goes to the question of why we need a new vaccine every year. Because these H and N proteins are very susceptible to change. Okay, they constantly are being changed. And one mechanism through which they can change is highlighted on this slide. Let's, for example, say one person is infected with 2 different flu viruses at the same time. One is H1N1 and the other is H3N3. Well, inside that body, as those viruses are reproducing, you can get a recombination event happening where a new virus gets created that has the hemagglutinin from this one and the neuraminidase from this one, and so we end up with a brand new virus, which is H1N2. And this happens very frequently, every year. And that's why every year there are new strains of virus circulating, and in February of every year, the CDC has to estimate which virus they think is going to be predominant for that flu season, and that's what we use in our vaccines. Okay? And so for this year, the 18-19 flu season, our trivalent influenza vaccine has 3 different antigens in it. It's got 3 different influenzas in it. There's an influenza A, which is an H1N1 strain. There's an influenza A H3N3 strain, and there's also an influenza B. And so that's the primary vaccine, this trivalent vaccine. There's also a quadrivalent, which has a 4th virus in it, and that's another influenza B from the Yamagata lineage. Yes, question.
Are you saying H3N3 on purpose? Do you not notice that it's H3N2?
I apologize, H3N2, yeah. That was a typo in my voice. Yes.
So why don't we just get a vaccine for all of the H's and all the N's?
So the question is, why don't we just get a vaccine for all of the H's and all of the N's? As with a lot of things I'm talking about up here tonight, it's not as simple as what I'm saying. There are a variety of different H1s and a variety of different N1s. So there's not just one particular type. So it's, again, more complicated than what I'm saying, and it wouldn't be possible to do that.
Okay.
Yeah. Yes?
Ronan, can you talk to the audience a little bit about the timeline that the CDC has in order to make these selections?
Yeah,
I think that might surprise people, how many months in advance they're making these decisions.
Yeah, so the decision is typically made in February for the following flu season. So like I said, the vaccine, like I'm encouraging you all, it's week 39 now, I'm encouraging you to get the vaccine now. But it was last February, in the middle of last year's flu season, that the CDC had to make a decision about what 3 or what 4 viruses were going to go in here. So that's a tough call, right? We're not even in week 40 yet. And they had to make a call way back in February about what 4 viruses they were going to pick. Do they get it right all the time? No. Do they get it right most of the time? Yeah, pretty close. But it's guesswork, right? And they use data like what strains are circulating in other countries in the world, in the southern hemisphere. But there's a lot of guesswork that goes into this as well. Yeah. Yes?
Two-part question. Live viruses or dead viruses? And if they're dead, then how does it work?
So the first question is, in the flu vaccination, is it live virus or dead virus? And if it's a dead virus, how does it work? The answer to that is both. There are flu vaccines that have both live virus and dead virus, and there's actually flu vaccines that are subunit viruses that just have the hemagglutinin portion. The dead virus, how that works is, a dead virus will still work as an antigen, and our immune systems will still recognize it and still start to produce antibodies against it. It doesn't have to be a live virus. If it's a live virus, some people would suggest it might work better because that virus is alive, the immune system can recognize that and develop antibody against that, but they both work pretty well. Yes. Oh, sorry, you had a second part of your question? That was it? All right. Yeah.
You got real close to what I wondered about, but I need a little more. I wondered if the customization each year is really just choosing these combinations, H1, N1, and so on, or if there's customization within one of those. And I think you suggested there's actually customization
Yes.
within.
Yeah, so the question is it as simple as picking H1N1 or is there customization within H1N1? And the answer is yes, absolutely. Within that H1N1 group, there are sublineages and subspecies, if you like, and that's really more the difficult question. Typically, every year the flu vaccine is going to have one H1N1 strain, one H3N2, and one influenza B. But it's which particular lineage within that group, that can be the difficult question. Yes?
So in Ohio versus in California, there might be 2 different kinds of flu going on.
So how does the CDC decide? Does it decide state specifically or
No.
all across the country?
So the question is, if you've got a influenza in California and Ohio, does the CDC make different vaccines for different regions? No, it just wouldn't be practical. It wouldn't be possible, right? So you can't just pick in February different vaccines for different states. So they make one. They make a call, and that's the same vaccine for the continental United States. And by and large, you tend to see the same strain circulating throughout the country. But again, some years it doesn't work as well as others. Yes?
So the people who are getting sick who have had the shot, do they have a different strain than the shot they got?
So the people who are sick who get the shot, is there a different strain than what they got, right?
Yeah.
So the answer to that can be yes and no. So when you get the vaccine, the idea is that it's going to prevent you getting the disease, right? And so if you get it, then maybe you just got a strain that you weren't immunized for, right? Or it could be that, the strain you got is a close match and you developed some immunity, but not enough to prevent you getting sick, right? So maybe it protects you. In many cases, the vaccine can protect you from getting sicker, or it can prevent you from dying, right? And so you might get really, really sick, but if you hadn't have got the vaccine, you might have got a lot sicker, or you might not be getting better. So there's a lot of that plays into it as well. All right, one more question.
I just have a quick question. So, is there a relative half-life of said strains, and is there a way to track the strains?
Is there a half-life of?
Like the strains, like different strains of viruses and stuff like that, and can you track them back to where the origin?
Yeah, many viruses. So can you track viruses back to their geographical origin? Yeah, in many cases they've been able to do that. Yeah, for sure. Especially with some of the more modern techniques that we have right now. Okay, I'm going to skip ahead, and I am going to get to the somewhat controversial portion of the talk that I was going to talk about today, and that is the vaccine controversy, which I'm sure everybody has heard about. This connection between vaccines and autism. And this sort of started back in around 1998 with the publication of this paper by this man. This is Andrew Wakefield, and he was a British former surgeon and researcher. And he was the lead author of his study in 98 that suggested that there was a link between the administration of the MMR vaccine and the development of autism in children. His conclusions were based on the fact that parents of 8 of the 12 participants in the study thought that there was a temporal link between when their children developed autism and when the MMR vaccine was administered. So any of you in the room who are researchers are going to look straight away and say, that's a pretty small sample size, right? And only 8 of the 12. And again, this data was based on parents' perceptions of there being a temporal link. But this really is the paper that started this, that it was the genesis for this controversy. Wakefield himself was attacked from the beginning, right? Other researchers published papers that said this is clearly not true. He defended it. He published a paper again a year later. And on this, what he's looking at is the approximate date when the MMR vaccine was introduced, and then looking at the number of cases of diagnosed autism in both the United States and the UK. And so he defended this argument, and he said that in fact the MMR vaccination was responsible, was the primary driver causing this increase in autism. So what is in this MMR vaccine? It was licensed in 71. In 1989, they decided that a second dose was good because it would increase the antibody circulating. And the vaccine contains 3 attenuated virus strains. So now you all know you can have live attenuated viruses in your vaccine, and that's exactly what the MMR vaccine is. And so these 3 viruses are measles, mumps, and rubella. And this vaccine is extremely efficient, right? Look at this chart of the number of measles cases in the US going back into the 40s, 50s, and then boom. Basically, measles doesn't really exist anymore, and there are a large number of physicians in the US who have never seen a patient with measles because of the efficacy of these vaccines. And so Wakefield came out with this idea that vaccines cause autism, and literally since that publication in 1989, there have been hundreds of studies that have shown no link between MMR and autism. Andrew Wakefield's study is to date the only publication that has ever shown a link between MMR and autism. The CDC, World Health Organization, all of these other institutions have shown that there's absolutely no link between these. So what's going on here? Where did this link come from? Well, in Wakefield's study, he suggested that it was this triple combination. By giving these 3 vaccines at the same time, it wasn't good for the immune system, it maybe overwhelmed the immune system, and this is what led to autism. And he suggested in his paper that individual vaccinations spread out over time may be a better approach. What he failed to disclose at this time was that he had just filed a patent for a single measles vaccination, okay, which represents a huge conflict of interest for somebody who's now discrediting this triple combination vaccination. The other thing was some of the parents in this study that participated were recruited by lawyers who were actually actively suing the manufacturers of the MMR vaccine, okay? And so they were recruited to this study, and these lawyers really wanted some parents that would come out and make some disparaging claims against the MMR vaccine, and that's, funny enough, what they got. Wakefield's study was also partly funded by the same lawyers. They provided about £430,000 for this study, all of which represent huge scientific conflicts of interest. So in 2005, Wakefield was sued, and I just want to read you a couple of excerpts from the judge's conclusion at the end of this trial. Number 1, he knew that his own laboratory's tests dramatically contradicted his claims. Wakefield knew that what he claimed in his paper was false, and this has been shown in court. Number 2, he repeatedly failed to disclose conflicts of interest, applying for a patent which, if effective, would have been commercially very valuable. It was going to be extremely valuable to him if the MMR vaccine was taken off the shelves. Number 3, he carried out research tests on vulnerable children outside the terms or in breach of the permission given by an ethics committee, essentially abusing children. He did things to children in the study that was not approved by any board. And then finally, he has been unremittingly evasive and dishonest in an effort to cover up his wrongdoing. To this day, Andrew Wakefield has not admitted that this study was falsified and was wrong. In 2010, he was found guilty of all charges. The Lancet have retracted the paper. If you try and download it, that's what you're going to find. But the result of this has been vaccine fear, okay? Unvaccinated children, and this fear and this perception that vaccines are bad. On top of that, think of the hundreds of thousands of hours of researchers and money that's been wasted on all of these follow-up studies trying to show a link between something that doesn't exist.
Is he in jail?
He is not in jail. He is thriving, and he is a poster boy for the anti-vax community in the United States. So why are we talking about it? We're talking about it because of celebrities who latched onto these ideas and decided to promote them. Sadly, it's not just celebrities that make movies. There's other celebrities that have latched on to these ideas as well and who put them forward in various different forums. So what are the arguments? What are the arguments from the anti-vaccination community? Well, unfortunately, the difficulty when it comes to talking about this is the goalposts keep changing. The anti-vaccination, people that believe that vaccination is wrong, their reasons for believing it's wrong always change. Whenever something gets debunked, some other idea comes along that is now the idea. And so here are just kind of 4 examples. Additives, right? There's toxins in the vaccine preparations. You know, there are a variety of different things that have been suggested: antifreeze, ether, aluminum or aluminium, fetal tissue, antibiotics. But the biggest one that we hear about is thimerosal, okay? This was the first thing that the anti-vax community latched onto. It's got mercury in it, so it's got to be bad. This was what was proposed to cause autism. The US began removing thimerosal from vaccines in '99. It's removed from all childhood vaccinations since 2001, with the exception of certain flu vaccines have it. But, you know, surprisingly, the number of autism cases have continued to increase. So clearly thimerosal is not the problem here. You've got a variety of conspiracy theories. It's all down to Big Pharma and the doctors. They're in it together. They're all making money in these vaccines. That's how they do it. It's just a big conspiracy and it's not real. You've got the natural is better approach, right? Things like natural immunity is better than vaccine-based immunity. Well, this sort of misses the point of vaccination, right? If you got to get a disease to get immune to it, that sort of misses the whole point. An infant immune system can get overwhelmed and can't handle all these vaccinations at the same time. Well, an infant is exposed to thousands of bacteria and viruses every day in and on their bodies. The immune system is designed to be exposed to multiple antigens at the same time. So again, this argument doesn't make any sense. And then you have the kind of crazy stuff like, well, the vaccines just don't work, right? Better hygiene and sanitation are the reasons that these diseases have declined. It's nothing to do with vaccines. We don't need them because the infection rates are already so low. Well, they're already low because of vaccines, right? That's kind of the point here. And then, of course, vaccines aren't worth the risk. okay? And again, I think that this has been conclusively debunked because the risks of getting these diseases are vastly, vastly bigger than the risks associated with the vaccines. So I'll finish up very quickly. I have like 2 slides or a couple of slides left with my personal, am I good? I can see Sarah getting closer here. There's one argument that you'll hear put forward repeatedly that I think takes some explaining, and I want to take a couple of slides to talk about this. And it's kind of summed up by these 2 quotes. It's my right to choose not to vaccinate, or I'm going to delay vaccination until my child is 2 years old. And these can seem quite reasonable, and I can understand these, and I've spoken to people, even members of my own family have come to me and sort of suggested these things. And there's one important thing that I want to bring up that sort of, kind of negates these types of arguments. And that's a concept or a process known as herd immunity. And how this works is, if enough people within a population are vaccinated, roughly 83 to 85%, if enough people in a population are vaccinated, the entire population become immune to that particular disease, right? So the microbe responsible has trouble spreading through the susceptible population. And it's sort of highlighted on this little cartoon diagram in here, okay? Most of the population are vaccinated. Here we have a non-immunized, sick and contagious person, but it's hard for the bacteria or virus to spread through the population to these unvaccinated people just because of the protection of the herd, okay? The more people get vaccinated, even those people that aren't vaccinated get protected. And these could be the people that are too young for the vaccines, people with allergies, or that have underlying medical conditions. However, if the percentage of vaccinated people within a population drops below 80%, herd immunity no longer applies. And so your decision to not vaccinate your child doesn't necessarily just affect you. It's not just your decision. There are children that can't get the vaccines because they have weakened immune systems, or because they're not old enough to do it, or because they were born premature. And so a decision to not vaccinate your child puts those children directly at risk. And that would be my argument to that particular point. So, with that in mind, I think I still have some time left, and get vaccinated, folks, all right? And I'll take any more questions that you have. Shoot.
So, shingles can be caused from having chickenpox.
Correct.
And you can't get it if you don't have chickenpox, but if you get the chickenpox vaccine, can you still develop shingles?
So the question is regarding chickenpox and shingles. If you get chickenpox, you can get shingles later in life. If you get a chickenpox vaccine, can you get shingles? The answer is no. It's the same virus, and so if you're immune to it, you can't get it. Yes. Yes.
Have you ever convinced anyone that was against vaccines to change their...
Okay, the question is, have I ever convinced anybody who was against vaccines to get them? The answer to that is I don't know. I hope maybe tonight I did. But look, a lot of studies have shown that if 2 people have different points of view, you can shout at each other all you like. You're never going to change each other's opinions. In fact, all you're going to do is really entrench those ideas. I hope that by presenting, an argument like this in this type of a forum, where it's not, I'm not accusing anybody of anything. I hope I can kind of, show people why some of their fears are unfounded. That maybe that can help in some way. But I don't know. I would like to think that maybe I have. Yes. The body produces antibodies?
Antibodies and vaccines help. What is it that causes somebody who has a organ transplant to reject?
Okay. Yeah. So the question is, what causes somebody to reject an organ transplant, right? And it's the immune system, right? So, as I said, our immune systems are really good at telling self from non-self, telling your body from everything else. And that even goes so far as to tell your organs from the organs of another human. So there are enough differences there that your immune system can start to recognize, hey, wait a minute, these lungs are not your lungs.
But they keep getting the medication for life.
Right, so the medication would reduce the activity of the immune system to stop your body from rejecting the organ.
But it doesn't develop it.
It doesn't...
Develop
And that's the idea, is that the medications will prevent your immune system from developing the antibodies against the lungs and the lung tissue that would ultimately cause a really bad reaction that would require you to take the lungs out, right? But of course, that could come with downsides because if you're on an immune repressors for life, you're more susceptible to infections. Yeah. Yes.
What about the super vaccine, the super flu vaccine for seniors? How does that differ from the regular flu vaccine?
Yeah, I was looking at this just before I came over, and I don't know 100%, but I think it's a high dose vaccine. I think it gives a larger dose of each of the viruses, and so it just stimulates a larger immune response and a quicker immune response. That's my understanding. I can't swear to that. I don't know exactly about each one of them, but I think that's what it is. Yes.
I just wanted to say something. If you get a vaccine, how many of you have ever had a vaccine and you don't feel really great afterwards? Right? It's not because you're getting the virus. There's a
Right.
compound in there called an adjuvant, which basically says, hey, immune system, let's react to this. It sort of prepares. That is very often what you're reacting to. If you have that reaction, very often
Okay.
there are different
Do you have one in
types
mind?
of vaccines that
Do you have a question in mind? Is there somebody I can
you
listen?
tell the doctor you've had that reaction. So in subsequent years, you can have a different vaccine. You're not actually getting the flu unless you've already been exposed to the flu. You're reacting to one of the compounds in the vaccine.
Yeah.
Sorry, being from a pharmaceutical company, I needed to throw that out there.
No, it's a great point, and you get that all the time. Is like, well, I got the flu shot, and then a week later I got sick. Okay, you didn't. It's as Roxy said, it's probably a response to the adjuvant in there, but you might have just got a cold the week after you got the flu shot, right? It's just most of the time it's a coincidence, right? It's not actually directly related. Yes.
The Nobel Prize in Medicine this year was awarded for the cancer immunotherapy techniques that were developed by two scientists, I suppose, a few years ago. Is that basically then an award for finding a vaccine against cancer?
Yeah, so the question is about the Nobel Prize in Medicine this year, which sadly eluded me again. Maybe next year. And so it was for cancer immunotherapy, right? And so yeah, this is the idea that you can train the immune system to target antigens that are present within the cancer. And this is a hugely hot topic in cancer research right now. And it essentially works on the same principle as what I've talked about here today. You can train those cells in your body, those adaptive immune system cells, to go after and kill the cancer tumors. Okay?
Do you see an anti-cancer vaccine?
It's possible. It's possible, yeah. Okay, last question.
Is there any way you can say in a layperson's manner why it is with cancer you don't have the same fundamental reaction going on in the body? Recognizing it's not right, it's foreign, go get it. That doesn't seem to be the case.
So as a non-cancer biologist, I'll give you my lay answer, which is probably a lay answer. My understanding of cancer, which is quite poor, is that cancer is such a broad term. So the question is, why is there not? The question was about why doesn't the immune system work as well against cancer? So two answers to that. One, it actually does. And so I think I've read an estimate once that on average, the cells in your body become cancerous. You get like one cancer developing every day in your body, right? And that cancer is destroyed by your immune system. So on the one hand, your immune system is actually very good at doing it. And on the other hand, cancer is just such a broad term. Prostate cancer is not the same as breast cancer, is not the same as brain cancer. There are so many specific, unique things to each cancer that it's very hard to kind of draw any broad conclusions as to why the immune system gets evaded by a particular cancer. But I think that the answer to focus on is that your immune system actually really generally does a great job of taking care of cancers before they even really get going. Yeah.
And with that, I would like to thank Ronan again, and I'm sure he will.
Yep, I'll stick around for more questions.